Hello and welcome to Fluid Mosaic Membranes Part 1. Okay, this is Chapter 4. So, as we've learned in prior chapters, a cell is the basic fundamental unit of all life on Earth. It contains numerous important structures and components that function together, with all the other parts interacting with each other. And as you know, the cytoplasm is the intercellular space within the cell containing all of these structures and components. So cells are often likened to a tiny metropolis, right, or a city. And like any town, a cell needs to create boundaries and to protect itself. So not much inside is static or stationary, including the cell membrane that surrounds it. All cells have a fluid but defined cell membrane that encircles the cytoplasm, like a city would have a moat, if you will. So in this way, a cell membrane provides a barrier to separate the cytoplasm from the extracellular space. Things are brought in and things are shipped out, but it would be dangerous to let anything pass through. Therefore, we also describe a cell membrane as selectively permeable, or only allowing certain substances to pass through the cell membrane. A cell membrane, however, is made up of a few more sophisticated parts than a moat and is constructed from entirely different materials. So the cell membrane is primarily composed of a phospholipid bilayer, cholesterol, proteins, and carbohydrates. So though the parts are different, it's easier to think about how the membrane functions by comparing it to the protective moat around a city. The composition of the cell membrane is best described by the fluid mosaic model, okay, where different parts of the membrane can float in a fluid-like space. So like it sounds, this model suggests that the cell membrane is dynamic or moving and asymmetrically scattered with different pieces, such as phospholipids, cholesterols, proteins, and carbohydrates, and that these different parts of the cell membrane float in the fluid-like space, moving laterally throughout the membrane. Oh, and additionally, the selectively permeable nature of the membrane allows the cell to communicate with other cells by cell signaling. Okay, so in chapter 1, you saw that all living cells are surrounded by a very thin membrane, the cell surface membrane, which controls the exchange of materials like nutrients and waste products between the cell, other cells, and its environment. Okay, they enable cells to receive hormone messages. And it's important to understand the structure of the membranes if we're going to understand how the functions are achieved. Okay, so the first thing we're going to talk about is the phospholipid. So if you start to think about what a mosaic looks like around a cell, right? If you're thinking of the fluid mosaic model, we'll start with one of the more popular components of the cell membrane, which is a phospholipid. The phospholipid forms the phospholipid bilayer. And as a refresher, this is the two layers of phospholipids that give us a membrane with hydrophilic heads oriented towards the outside and inside of the cell where there is an aqueous environment. And the fatty acid hydrophobic tails of the phospholipids don't like to get their tails wet, so they face each other, creating a hydrophobic region inside the bilayer. And a function of that bilayer is to create a distinct hydrophobic region, which is essential in both keeping fluidity inside the cell and moderating who can get through the selectively permeable membrane to get into the cell, right? So in later lessons, we'll talk about who's allowed through and when. So from phospholipids, little bags can be formed inside, which chemicals can then be isolated from the external environment. And these membrane-bound compartments are cells and organelles. They form a single layer with their heads in the water because they're polar, like we just said, and their tails projecting out of the water. Okay, and we know that the term polar refers to the uneven distribution of charge which occurs in some molecules. Okay, and I want you to think about that again. I want to kind of go back to that and make that point again. So it's a hydrophobic and hydrophilic bilayer. Okay, and again, they form a single layer with their heads in the water because they are polar. Okay, they are attracted to water, right? Because water is has polarity, and so therefore is attracted to things that are also polar. And then the tails, which are non-polar, okay, don't want to be near the water. Okay, so if the phospholipids are shaken up with water, they can form stable ball-like structures in the water called micelles. Okay, and this is an important one. Really take a minute, pause the video, really look at these diagrams that I have here. Okay, all the hydrophilic heads face outwards into the water, shielding the hydrophobic tails, which point in towards each other. Okay, alternatively, two layered structures called bilayers can form in sheets. It is now known that this bilayer, right, this phospholipid bilayer, is the basic structure of membranes. The double black line you can see here through this electron microscope is thought to show the hydrophilic heads of the two phospholipid bilayers. Okay, the pale zone between is the hydrophobic interior, and the bilayer membrane is about 7 nanometers wide. Okay, and contains embedded proteins. So, coming back to the fluid mosaic model. The membrane is described as fluid because both phospholipids and proteins can move about by diffusion. Okay, the bilayer has fluidity, and you can think about it. You can think about the viscosity that is similar to olive oil. Okay, which this means that they can move sideways in their own layers. Okay, proteins move like icebergs in the sea. The word mosaic describes the pattern produced by the scattered protein molecules when the surface of the membrane is viewed from above. Okay, so here you can see the membrane structure according to the fluid mosaic model. So you can see those, the cellular membrane, the phospholipid bilayer. Here are the hydrophobic heads in the center. They're facing each other. Okay, which should tell you that water is going to be on this side of the cell, right, in whatever matrix it's floating in, and then cytoplasm is inside the cell. Okay, you have proteins, which we're going to learn all about those in a minute. Okay, and you can see they're moving sideways through this, okay, which is, again, why this is called a mosaic, is there's these different components of the cell membrane that are floating around. And again, this is the consistency or the viscosity of olive oil, and these things are sort of floating around. Okay, here's a model that you should definitely study. We're going to talk about the glycolipids, the carbohydrates, the proteins, glycoproteins, cholesterol. We just talked about the phospholipids and the transport proteins. Okay, so the membrane is that double layer bilayer. And the individual phospholipid molecules move about by diffusion within their own monolayers. The phospholipid tails point inwards, and the heads face the aqueous medium. Okay, some of the phospholipid tails are saturated and some are unsaturated. The more unsaturated they are, the more fluid in the membrane. So this is because the unsaturated fatty acid tails are bent, and and therefore fit more closely to fit together more loosely. So fluidity is also affected by a tail length. The longer the tail, the less fluid in the membrane. So as temperature decreases, membranes become less fluid, but some organisms which cannot regulate their own temperature, such as bacteria and yeast, respond by increasing the proportion of unsaturated fatty acids in their membranes. Some of the phospholipid tails are saturated and some are unsaturated. The more unsaturated they are, the more fluid in the membrane. And this is because the unsaturated fatty acid tails are bent, so they fit together more loosely. Fluidity is also affected by tail length. The longer the tail, the less fluid in the membrane. So as temperature decreases, the membranes become less fluid, but some organisms which cannot regulate their own temperature, like bacteria and yeast, respond by increasing the proportion of unsaturated fatty acids in their membrane. So two types of proteins are recognized according to their position in the membrane. One is the extrinsic or the peripheral protein, and this is found on either the inner or outer surface of the membrane. Now, intrinsic proteins or integral proteins are ones that are found embedded within the membrane. Okay, intrinsic proteins may be found in the inner layer, the outer layer, or most commonly spanning the entire membrane, in which case they are known as transmembrane proteins, right? Because they're moving, potentially moving particles over or through the membrane, so transmembrane. And in transmembrane proteins, the hydrophobic regions which cross the membrane are often made up of one or more ahelical chains. Okay, so intrinsic proteins stay in the membrane due to its hydrophobic and hydrophilic regions. The hydrophobic regions, made from hydrophobic amino acids, are next to the hydrophobic fatty acid tails, and so then are repelled by the water environment on either side of the membrane. And the hydrophilic regions, made from hydrophilic amino acids, are repelled by the hydrophobic interior of the membrane, and therefore face into the aqueous environment inside or outside the cell, or line hydrophilic pores which pass through the membrane. Most float like mobile icebergs, although some are fixed like islands to structures inside or outside the cell, but do not move about. So they're really a part. Proteins are just really an important part of the cell membrane. The hydrophilic ends, so you can see here in this model, of the molecule exposed to the aqueous solutions on either side of the membrane, and then the hydrophobic regions that completely span the hydrophobic interior. Okay, so many are bound to intrinsic proteins. Many extrinsic are bound to intrinsic, and some are held in place by binding to molecules inside or outside the cell. So if you look at this model here, you can see that the extrinsic proteins are on the outside of the membrane. And then in this one here in the middle, okay, you have this extrinsic protein, and then you have an intrinsic protein that is bound to it. Okay, many proteins and lipids have short branching carbohydrate chains that are attached to the side of the molecule that faces the outside of the membrane, thus forming glycoproteins and glycolipids. So the total thickness of the membrane is about 7 nanometers on average. Molecules of cholesterol are also found in the membrane. Okay, so proteins' roles in membranes. So there are transport proteins, which are hydrophilic channels. Okay, they move ions and polar molecules. And you know that because they are hydrophilic, right? So they like water, and remember, water is a polar substance, so a hydrophilic channel would be okay with ions and polar molecules moving through. There are enzymes, which are on the plasma membrane on small intestine surfaces, which hydrolyze disaccharides. There are proteins for photosynthesis and cell respiration, E-transport chains in mitochondria membrane, and the thylakoid membrane of the chloroplast. And there are receptors which bond to the antigen and begin communication pathways. Okay, so you see those transporters, receptors, enzymes, etc. Okay, so make sure that you're studying the boxes and the pop-outs in the book. These are going to have all of those little bits of information and specific examples that you really need to understand in and out. Okay, thank you, and feel free to watch the next video.